again. In the last video we derived the Hardy-Weinberg equations, and now we'll look at how to use those. And remember, this is in a large population with no particular changes and no natural selection to change the gene frequencies of the organisms. You can see the organisms I've chosen, because they're easy to draw, are fruit flies. So we're looking at the same sort of thing that Thomas Hunt Morgan did in the early 20th century with his experiments with the genetics of fruit flies. And so we can see here, some of them had red wings, some of them have white wings, and they're the two phenotypes. And what we want to do is have a look at what kind of genotypes would create those two phenotypes. And maybe by some process that Hunt Morgan did, we might get the idea that the capital A, the dominant, codes for white wings, and of course the lowercase a would therefore code for red. And therefore we've got the situation here where we've got 4 out of 10 of our population, that is 0.4, being the homozygous recessive. Because of course to have red wings, they're not permitted to have one of the capital A's. And so we know that the frequency of this genotype here, which gives rise to the red-winged phenotype, is 0.4. And that means the rest of them, the other two genotypes, the homozygous dominant and the heterozygous, must add up to 0.6. Now, when we look at this bunch of fruit flies here, then we have a look at the idea of how to work out the frequency of the two alleles. And of course it's tempting because you see... I have no idea what just happened then. Because of the smaller number of the recessive, it's tempting to think that the recessive allele has a lower frequency than the dominant. Well, let's see if that's the case. So we've got f of q squared. So remember, this here, this equation, if we rewrite that using these alleles, then it's the frequency of homozygous dominant squared plus the frequency of these, and remember that's 2 of those, plus the frequency of these equals 1. All right, so, and that corresponds to how it happens when we do a Punnett square of a mating of 2 heterozygous individuals. 1/4 of them homozygous dominant, 1/4 homozygous recessive, and of course 1/2 of them, 2/4, being heterozygous. Oops, I missed that out. So we have a look at how we can do some math here to work out the frequency of the alleles. So we know that q squared, this one, is 0.4. So what we can do now is work out this, because we know that q squared is 0.4, we know that q is the square root of 0.4. And if I work that out with my handy calculator here, which I already have to be honest, that equals 0.632. So the frequency of q is 0.632. And that means if we work out p, the frequency of p, is 1 minus 0.632. And that, of course, is going to be less than 1/2. And that's 0.3, let me check my notes, 68. And so there we get the idea that actually the frequency of the capital A, the dominant allele, is actually lower than the frequency of the recessive.